All. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So the question is that why in some people polyethylene glycol or PEG in vaccines may cause allergic reactions, and should the vaccine advisory be that, hey, if you have the past allergic reactions, then be careful, or something more than that? So that is something that I would like to discuss. And I'm not going to be discussing the PEG use in various products. Instead, what is the mechanism which causes the allergic reactions, which causes the autoimmune responses to PEG in some people? So let's start. So these are our gifts for humanity. And if I can very quickly show you, this is drbeen.com. In the description, there is a link to the premium account for for the one-time price of 2 or 3 lunches. So take advantage of that if you can. Here is the article, Polyethylene Glycol Immunogenicity, Theoretical, Clinical, and Practical Aspects of Anti-Polyethylene Glycol Antibodies. I have never seen such a comprehensive article before, and I would only be able to go through those parts of it that are related to the vaccine and nanoparticles, which they have discussed here. So somewhere little down, as you can tell, it is an exhaustive content. So liposomes and nanoparticles. So that is the main thing, but I have to go through all the context of it. So let's start a discussion with my drawings. So the first question that I would like you to have in your mind, when the vaccine is injected, that is lipid nanoparticle and has polyethylene glycol in it. And I'll explain in a few minutes that why is it in there, but has the PEG in it. What are we expecting from our body? How are we expecting the body to respond to this vaccine? How are we expecting our immune system to respond to this vaccine, lipid nanoparticles? That is a question. There are 2 basic answers. One answer is to react, our immune system to react to the lipid nanoparticle, and the innate arm picks it up and processes it and starts a quiet arm. That is one answer. The other answer is lipid nanoparticle needs to get into normal commoner cells, for example, muscle cells or other cells present in that vicinity of where it is injected. Then the mRNA needs to come out of the lipid nanoparticle. That mRNA needs to make spike proteins, which would then be shredded and presented on the surface, and that would cause the immune system to respond to the spike protein antigens. What do you think? Which answer is the correct one? The second answer is the correct one. We do not want the lipid nanoparticle. The lipid nanoparticle, here, if I draw this, lipid nanoparticle has the mRNA in it. We don't want this thing to be picked up by immune cells. Why? Because this particle has to yet enter a cell, provide the mRNA to the cell, and then have the cell make the spike protein. We want to create immunity against the spike protein, not the particle. So then, if you and I are manufacturing this drug or this vaccine, we will try to do everything in our power to make sure that immune system does not look at it. Immune system does not pick it up. Immune system does not process the particle. Instead, the particle is given a way so that it can enter the muscle cell or whatever is the cells in that area. So that is the basic context. Let's start. Lipid nanoparticle on why does it cause allergic reactions? Here is the summary of it. Number 1, PEGylated. So those drugs that have lipid nanoparticles in them, that have polyethylene glycol in them, are called PEGylated drugs. For example, Moderna vaccine and the Pfizer-BionTech vaccines are PEGylated vaccines. There are drugs that are PEGylated as well. So PEGylated lipid nanoparticle based vaccines can cause anaphylaxis usually by the second dose. That is one message to keep in mind. Second, even the first dose could have a reaction if someone was taking other drugs that were PEGylated. So then you would say, are there other drugs that are PEGylated? Yes. And this has been for decades. There have been drugs that have polyethylene glycol in them. And I'll again explain why. But let's look at some of these drugs. So if you or someone you care for, some friend, even strangers, if they are using these drugs, they should be a little more careful. And this is what I thought should have been from the healthcare authorities to say those persons who may have been taking PEGylated drugs, they may have a chance for reaction. Just like those who were previously had allergic reactions. So look at this. This is clinically used PEGylated protein drugs. So the drugs that are PEGylated or with which the polyethylene glycol is attached, these drugs are in two categories, protein and non-protein. And all of that would become clear. Let's look at the drugs names first, or drugs types first. And I'm not going to go over all of that. What I'll do is this. If I turn towards scroll it to the right, you would see that these are the drugs that are given to patients of neutropenia, hemophilia, phenylketonuria, very important and very common, severe combined immunodeficiency, melanoma, gout, rheumatoid arthritis, anemia, acromegaly, hepatitis, neutropenia, hepatitis C, leukemia, severe combined immunodeficiency. Now here, those patients that have the immunodeficiency, for them to mount in an immune response or allergic response is less possible. So even if they had been taking PEGylated drugs, the vaccine may not pose that kind of allergic reaction danger to them because their immune system is already not optimally responding. So these drugs have polyethylene glycol in them. Then here is another table, clinically used PEGylated non-protein drugs. So again, I'll explain why protein and non-protein, but these are non-protein drugs. And if again in here, if you see the last two lines are the two vaccines, Moderna's vaccine and BioNTech's vaccine, Pfizer's vaccine. And if you see here on the right side, the kind of disease, cancers, macular degeneration, constipation, cancer, amyloidosis, and COVID-19. These are some of the drugs. And if somebody is taking these drugs, it is interesting and important for them to know. And I would have thought, once again, I'm repeating myself, that healthcare authorities would have said, hey, if you have been taking PEGylated drugs, be careful or talk with your doctor. Okay, going back here. And if this has raised a question in your head that why, I would explain that. So even first dose could cause a reaction if someone was taking PEGylated drugs in the past. Then there is another issue, and that is, and I think, and this is author's conclusion as well. So that is that after massive vaccine campaign, a large number of people have received the lipid nanoparticle based vaccines, including me, and they may have developed anti-PEG antibodies. They may have developed immune reaction to these to the polyethylene glycol, which means such folks in the future will be at risk of getting another vaccine, another drug, another therapy that has PEG in it and getting a reaction or getting the immune system to flare up, immune system's response to flare up, inflammation to occur. Now, you know that with allergies do not happen to everyone. About 20% of the population has allergies. Other people are fine for the same, for example, pollens or cat hair or the dog hair or the dander or the dust. So it is not necessary that this happens to everyone. But what is interesting and important is that this will happen to some. And we should be aware of that. Why I'm repeating this is I never, in these 2 years, I never heard a message from anywhere to say polyethylene glycol may be causing allergic reactions. So if you have been taking these drugs, be careful. Or if you're going to take those drugs in the future, be careful. Okay, so this is a summary that PEG can cause allergic reactions. Now, the question why is what I'm going to go in the detail of. So if you just wanted to know what can happen, we've done. Now, how can this happen? So let's go step by step. This is ethylene. So there are 2 carbons and there are 4 hydrogens. CH2, CH2. This is ethylene glycol. Again, there are 2 carbon, but you can see that there are, in addition to these 4 hydrogen, there are 2 OH added as well. This is ethylene glycol. Now, if you take ethylene glycol, if you look at this part of the diagram, if you take ethylene glycol and repeat that molecule and maybe combine that molecule with other things, and that becomes polyethylene. Polyethylene means many ethylenes. Glycol, so this is actually polyethylene glycol, and then it is connected with some other things. What are the other things? It depends what kind of immune response you want or you don't want. So the first question, why do we use this thing? We use polyethylene glycol because it allows the let me actually show you the table. So here, these are various kind of pegs, and if you see here, there are various lengths of this, and then the viscosity diameter changes. What that means is that these molecules can be long or short, branched or not branched, and they can adhere water around them. So if I wanted to say that, hey, I want to have my personal space, and my personal space is my arm's length everywhere. How can a molecule have a personal space of an arm's length? That is, if you attach polyethylene glycol to it, then that molecule would have a viscosity diameter that is bigger than the molecule itself. And now that molecule can move around in our body safely because it is wrapped in a packaging that is protecting it and creating a viscosity diameter around this. So let me show you another example. Down here, these blue guys, these blue lines are polyethylene glycol. And stuck with them are these tiny little proteins. These are the ones that are using this polyethylene glycol. And here, this little diagram where there is a part here, this is actually lipid nanoparticle, and these blue ones are the polyethylene glycols mixed with it. And now the question is, why? So we know now that there is polyethylene glycol. It can be of various lengths. It can be branched or not. It increases the viscosity diameter, creates a personal space around a molecule so other things do not bother it. What other things? Most importantly, immune system. We don't want immune cells to look at this product and react to it. This is sort of a stealth system for immune. Not very successful, but much better than many other molecules. So what does that mean? For example, in the case of vaccines, we do not want the lipid nanoparticle, and by we, I mean the vaccine manufacturers. They do not want the lipid nanoparticle to be picked up by the Kupffer cells in the liver. Kupffer cells are the macrophages in the liver. They don't want the particle to be picked up by them. Why? They don't want this particle to be picked up by any immune cell, because the point is that the particle should stay without going into the immune cells to go into the normal cells and make spike proteins there. Second reason for polyethylene glycol to be present with these vaccines is that we don't want immune system to attack this, not only just pick it up, but we also do not want the immune system to attack this thing. And again, I keep saying we, I mean the vaccine manufacturers. So what we do is we attach this polyethylene glycol around it, and that keeps the immune system away from it and away from attack. And why is the attack bad? So imagine if a macrophage picked it up, then what? Fine, macrophage would pick it up and it would present the components of this particle, not the spike protein. It would present the mRNA, or it would present the various lipid particles pieces here. That is one reason. That's useless. That's like eliminating the vaccine. You inject the vaccine in somebody to do some function, and the vaccine is already wiped out by the immune system. It's going to do nothing. The second thing, we don't want the immune system to attack and destroy and open these particles outside in the tissue. We want it to safely get into the cell to make spike proteins. If you break it outside, it's going to open up and the mRNAs are going to come out, and that would cause a different kind of immune reaction. That might even create anti-mRNA or RNA, anti-RNA antibodies as well. We don't want that. Okay, so what do? We decorate it with polyethylene glycols. So then, can this decoration, to try to safeguard this particle from immune system, create a problem of its own? Yes. So let's look at that problem now. So here, let's say we have a macrophage, and the macrophage picked up the polyethylene glycol particle. Once again, in theory, macrophages would do this less. And I would go to a second part of this discussion in a few minutes to explain that when macrophages and innate arm is not attacking it, then what other mechanism is possible to create the antibodies? So let's go through the classical mechanism. The particle is picked up, it is presented to naive T cells. Naive T cells will then go either the T helper 2 pathway or T helper 1 pathway. In case of the lipid nanoparticle or this particular polyethylene glycols, the pathway goes towards the T helper 2, which will mean that the B cells will become active and they will make antibodies. Now, here is a delicate point to consider. This whole nanoparticle is lipid. Polyethylene glycol normally does not cause any issues as well, because that is also carbon and hydrogen, lipid-like thing. The B cells usually work with proteins. B cell bind usually to proteins. So here we have a gap. What we have given is lipids. What the B cells bind with normally are proteins. So we have a problem. But here is the thing. When the B cells get the interleukin 4 and 5, and the B cell has a polyethylene glycol based lipid nanoparticle or mixed lipid nanoparticle, connect with their IgD and IgM. B cells, when they are produced, they're naive B cells, they still make IgD and IgM. Those IgD and IgM are usually not released in the blood. Otherwise, we'll just have random antibodies all the time. Instead, these antibodies are connected to their surface. These antibodies act as B cell receptors. Now, these receptors can sense the polyethylene glycol or the lipid particles antigens on it, especially polyethylene glycol part. And if that happens, they cannot really process it, but they will start making more IgM. They will start saying, well, something touched my hand. That means there is something fishy going on. Some antigen is present. You know what? I will just make more IgMs. And this time, the IgMs they will make, they would start releasing it in the blood as well. This is called a weaker response. It's not a very robust response. The antibodies are not changing from IgM to IgG to E to A. They're just IgM. They also make IgD, but IgD is not very interesting at this time. So weak response. But there are antibodies now against the PEG, polyethylene glycol. Then what happens? These polyethylene glycol based antibodies or anti-PEG antibodies, they can go, number one, be present in the tissues. And when the next dose of the drug would come in, they would attack it. But more importantly, these can go and get connected on the mast cells and they can prime the mast cells. Mast cells are responsible for the allergic reactions. So the first time the priming would occur, these antibodies would go and get attached with the mast cell. The next time when this antigen is brought in, let's say the second dose, then the antibodies that are acting as receptors, when they connect with these polyethylene glycols, that would make the mast cell very angry because it would say, well, you came back again, and it would become degranulated. It would release histamines and many other things, which would then cause severe allergic reaction to a point that some people die as well. And we know that with the vaccine allergies, some people have died. So this is one, but this is a weaker response. This response here of producing antibodies may not be sufficient to have enough priming of the mast cell that the mast cell do this. But you may be thinking, well, okay, then we are out of the woods. We do not have a problem. We have a weak response, and that response is not sufficient to hurt anyone. No. Hang on. There is another response that is called Thymus independent antigen response. So let me explain this term for a second. When our body's immune system responds to protein-based antigens, then that is called Thymus dependent antigen response. Why? Because the T cells, they are trained in thymus. And T cells only work with the protein-based antigens. So whenever we have an antigen that is protein-based, T cells would come into play as well. When they come into play, they are called Thymus dependent antigen antibody response system, or they create that. So here we have a thymus dependent response system, and we looked at it before as well, and this is a weaker response. But in our body, we also have thymus independent antigen response system, or response to thymus independent antigen. That means antigen that doesn't have protein in them. Lipid nanoparticles do not have a protein in them. If they have mRNA to which the thymus dependent, which can be called thymus dependent antigen, that mRNA is kind of hidden inside the particle. We've done a lot of things to kind of create this structure. Still, this can be a thymus independent antigen, and let's see how does that work. So imagine there are a bunch of lipid nanoparticles and they have PEG on their surface. When the polyethylene glycol connects with the IgM and IgD on a B cell, but connects it in a large quantity, these B cell receptors, when they are tickled in a larger number, then the B cell says, man, there is a problem. It does not need to bring it in, the lipid nanoparticle. It does not need to break it down. It does not need to present it. It simply says, I'm going to make more IgMs. And that weak response starts, as we discussed before. However, here is the next part of the response. The innate arm can help this B cell without this classical pathway of activation. This classical pathway does not engage, and even then this cell becomes activated. How? So on the innate arm, we are aware that there are cells like neutrophils, dendritic cells and the macrophages and the myeloid cells. There are specific neutrophils as well that are called B cell helper neutrophils. There are many kinds of neutrophils. B cell helper neutrophils, what they do is they pick up the lipid nanoparticle. They digest that, they phagocytose it, they digest that, and then when they think there is something that is from outside and it is not good. This is despite that we have wrapped the lipid nanoparticle in the pegs and we are trying that immune system doesn't see it. Immune system is going to see it, some of it for sure. So these neutrophils that are B cell helper neutrophils, when they eat up this one, they start producing chemical substances that are going to encourage B cell to say, you become fully active now. You should, even if you do not have the whole classical pathway engaged, you should become active. Similarly, the other myeloid cells in the innate arm, they also do the same thing. They also engulf the lipid nanoparticle. They also break it down and, excuse me, and they also release more substances, chemical substances. All of these chemical substances, they go to the B cells, they attach with it, and the B cell is already feeling the heat. It's already getting suspicious that there's so many lipid nanoparticles. So they tell the B cell to become fully active. This is activation of a B cell by a thymus independent antigen. The question is, what are these chemical molecules? So for the medical folks here, the myeloid cells release BAFF. So these are B cell activating tumor necrosis factor family molecules. So these are TNF molecules, tumor necrosis factor molecules, that would allow the B cell to become proliferated, B cell to become more active and start producing antibodies. Then we have APRILs. APRIL is a proliferation inducing ligand molecule. What does that mean, APRIL? A, proliferation. Proliferation means the B cell would be asked to increase in number. So proliferation inducing ligand. Ligand simply means something that can attach with another thing. For example, in the case of SARS-CoV-2, the spike protein and the ACE2. ACE2 is the receptor, spike protein is the ligand. Ligand is anything that can attach with another thing. So APRIL molecules and BFF molecules, so not BFF, BAFF, they come and attach with the B cell. The result is B cells become proliferated and becomes fully active and does class switching to other immunoglobulin types and becomes proliferated and becomes fully active. The result is, as part of that proliferation and activity, IgM is released. So this is an additional step. This IgM that is released would activate complement system. We know that whenever antibodies are released, they would activate the complement system. When the complement system is released, there is complement sensitization, or the complement receptors are then attached to the B cells. This B cell in the future with the second dose will become even more readily proliferating. So B cell, you can say, this is not the right immunology term, I am using it. B cell becomes primed for the next time. So when the next time the same PEG arrives again, now that PEG can attach to the complement receptors, and this B cell will become active. So how are we activating the B cell? What is the nutshell of this mechanism? The polyethylene glycol is a thymus independent antigen, and we are still be able to create antibodies to that, IgMs, IgGs, and As, Es, because we can. Now, if I go back here for a second, this mechanism will not happen with PEG, or would happen in a very tiny way. So all of this, which is a classical mechanism, may not happen, but this other mechanism would fully engage, and we would once again arrive back in the same situation. So that is the talk what is the takeaway? I'm going to put the summary back here. I usually put the summary in the beginning, but sometimes folks say, hey, what was the takeaway? So the takeaway is this. Number one, doesn't happen to everyone, allergic reactions to PEG. Number two, and this doesn't mean that the vaccine only has PEG to have allergic reaction to. Maybe there are other components in the vaccine that we can have allergic reactions to, but PEG is known to do that. So the reason to have the PEG in the vaccine is to allow the immune system to not see the particles and allow the particle to enter the cells and do its job. The problem is that our B cells and the innate arm, they would still work and they would still look at the PEG and attack it. And if we become, a percentage of us will become sensitive to it, we would start making anti-PEG antibodies. And here is the important thing. We do not know how many people have developed anti-PEG antibodies. So let's say out of 4 billion doses given, let's say 500 million people have developed anti-PEG antibodies. Now, what is the possibility? There is actually no data for this. The data that we have is for another drug, excuse me, that is a PEGylated drug. And in that drug's case, it is in here in this article, they say that about 20 to 23%, if I'm recollecting the number correctly, that is the number of people who develop anti-PEG antibodies. So if we take that, then 20-25% of the all vaccine receivers who have received the lipid nanoparticle based vaccines with PEG in it, they would have anti-PEG antibodies. Ideally, there should be a study done. But anyways, now these folks who develop these antibodies, some of them are going to develop allergic reactions, not only to the vaccine's next doses, but also to the other drugs that they may need in the future, which have PEG in them. They might respond negatively. And then what is the confidence level that this would happen or not happen? I think it's a similar thing as allergies. Not everyone has allergies, but some have. So similarly, not everyone who develops the antibodies will have a problem, but some will. What is that percentage? It is still not known, but I think this should have been made clear to all of us as well. So here, once again, ending it, LNP with the second dose can cause a problem. Why the second dose? Because the first dose, as I showed you, would prime the immune system. The second dose, when it arrives, the immune system would just attack it. Even the first dose can cause the allergic reaction. And that is when somebody had taken or was exposed to PEG in some way and did not even know that they have antibodies against PEG. And now they got the first dose of vaccine had PEG, and the attack started. And the third is a massive number of people are now, if they didn't have PEG before, now a massive number of people are going to have anti-PEG antibodies. And that means in the future to such therapies, they are going to react naturally and negatively. So that is the discussion. I hope it makes sense. Let's see if there is any question. Otherwise, my request to you, please like, subscribe, and share. And there are some links in the description as well. If you like this work, then please support this work, because I really am hoping that I move away from YouTube at some point. It's not that I'm moving away, but I would like to have that independence, because there is censorship here. So if you'd like, one easy way to support this is that there is a link in the description to Drbeen premium content. So you get 1,000 videos for a very, very cheap price, very inexpensive, affordable price, probably 2 lunches, and access to 1,000 videos. And the second is you can use Buy Me a Coffee to support this work, or you can use PayPal to support this work, or you can become a patron. I really would love someone to become a patron or a Substack member. because then we can just continue to support this work. So with this, thank you very much. Easiest part, like, subscribe, and share. And in a few minutes, I will then join you again on the Cool Beans Cafe. So that is, if you go here to YouTube and you type Cool Beans Cafe Live, you will come across this. This is the Cool Beans Cafe. I'm going to put this link in the chat as well. And we will go to the Cool Beans Cafe and we'll do a quick chit chat, maybe even answer the questions related to this particular discussion, and see you soon. Bye-bye.